Okay, good afternoon. Good afternoon, everyone. So can you hear my voice, Fons? And
Yeah, yeah,
yes,
and
yeah,
Can.
okay, then. Good afternoon. So in this sunny and then a bit cloudy day, so we will have a presentation from this section about the herbarium and museum of the School of Life Sciences and
oh,
Technology
oh,
here. Um, yeah, I'm here as a speaker as well, and then I will would like to introduce
oh.
my college. Is there any problem? Okay, with the sound. Perfect. Okay.
There's an echo, so it's
Oh, echo. Okay, we will fix it first. Okay. I hope the voice is better now. Yeah. So on this schedule in our presentation, it will be presented by 7 person from Indonesian part and also with the Netherlands part. So the first presenter will be presented by Dr. Ruliyana Susanti from BRIN. And also next presenter is myself. And then with the experience to taking sample of the herbarium and also the museum who has many experience in the field. This is uh, Arief and will be the next presenter. And the last presenter will be from Netherlands. So we have our college Fons here. And also we have Liu and also we have Jens and Angelica here. Thank you very much for joining with our presentation. So I would like to give Dr. Luri on the stage. So yeah, we welcome Dr. Lily on the stage, and then she will present about 15 minutes about the Indonesian biodiversity. This stage. Yeah.
Hello, everybody. Good afternoon, ladies and gentlemen, colleagues, researchers, students, and partner in Indonesia and in Netherlands. Nice to meet you all. My name is Ruliyana Susanti from the National Research and Innovation Agency, or we know it as BRIN. Here is it an honor for me to be here to share some reflections on Indonesian biodiversity and how our collection serve as a living record of that richness. Since I'm not a native speaker, so I prefer to use the text. So this is how my professor previously teach me how to present in English. So we can keep the time and then reduce the mistake. Um, Ladies and gentlemen, our gathering today is very important as we advance into an era of rapid environmental change and technology transformation. The way we document it, preserve, and also uh, digitally manage biodiversity data will determine how effectively we can protect the biodiversity for the future. As someone working both in biodiversity research and biodiversity policy, I have seen firsthand how collections are not just archives. They are the scientific backbone of conservation, taxonomy, and sustainable developments. Next. As we all know, Indonesia is recognized globally as one of the mega diverse countries, home to an extraordinary range of ecosystems, species, and genetic resources. Our archipelago natural history has shaped by complex geological process, volcanic activity, plate tectonics, and climatic variations, creating diverse landscape and ecological zone. This has given rise to 7 major ecoregions for the National Development Plan, from Sumatra rainforest to Papua alpine ecosystem, each hosting unique flora and fauna. Such diversity is both a scientific treasure and management challenges. Every island, every ecosystem, and every specimen holds stories of evolutions, adaptations, and survival. And it is through collection that their stories become part of our scientific knowledge. Next. Indonesia. Next. So Indonesia geographical further amplifies its biodiversity with over 70,000 islands. Our nation stretches across the equator and connect two great oceans through the Indonesian Through Flow, or we know it as Arlindo, Arus Lintas Indonesia in Bahasa. This dynamic setting formed the core of the global coral triangle, home to the highest marine species diversity on Earth. Such natural connectivity also shapes how species move and evolve. Yet, ironically, this archipelagic nature also makes our biodiversity fragmented in knowledge, with many regions and taxa still underrepresented in collection and database. This is precisely why museum and herbarium collection are essential. This is why we are now gathering here. Next. Across the islands, Indonesia holds 22 recognized major ecosystem types. This is included in Indonesian Biodiversity Strategic and Action Plan, ranging from mangrove and sea grasses to mountain forest and alpine ecosystem. Next, each ecosystem contains species assemblages shaped by climatic, topography, and evolutionary history. This data is included in our recent biodiversity stocktaking book about explaining about what we have, what is our ecosystem, species, and genetic diversity have in Indonesia. Among all, Papua holds the most complete set of ecosystem types, yet the least informations for Indonesia regions. Next. Java, Sumatra, and Kalimantan are better studied, primarily due to historical expedition that laid the foundation to our herbarium and museum collections. However, Eastern Indonesia remains a vast frontier for discovery. In fact, many species description today still rely on specimens collected decades ago, reminding us on how vital well-preserved collections are for modern science. Collection, therefore, are not just repositories, they are time machines, allowing us to revisit and reinterpret biodiversity through the lens of new technologies. Next. 7 ecoregions across Indonesia host highly endemic species, with Sulawesi standing out as an evolutionary crossroad where Asian and Australian lineage meet. The ability to recognize endemism, the name and describe new species, or to assess conservation status all depend on our voucher specimen. Without them, species would remain invisible to science, policy, and conservation planning. Next. This is examples of documented of our endemic species in Sulawesi in the terrestrial and the marine regions. Next. Our herbaria, museum, and microbial culture collection are thus central to biodiversity governance. They provide variable physical evidence that anchor our biological data. However, their potential is often limited by accessibility. Many valuable collections remain undigitalized, stored in cabinets and boxes, known only to few researchers or experts. Beyond plants and animals, biodiversity extends to microorganisms. It is estimated that at least 1 trillion microbial species exist globally, yet less than 1% have been identified, including in Indonesia, maybe less than 1%. Indonesian microbial diversity is a large untapped frontier. Unlocking it requires whole genome DNA sequencing and integrative biobanking, which link genetic data to specimen-based record. Next, not only microorganism, but our genetic diversity also very important and yet not yet fully explored. Um, This intersection between biological collection and digital data marks the frontier of biodiversity science today. Digitalizations allow us to connect herbarium voucher, animal specimen, and microbial isolate into one integrated knowledge system. Next. In today's context, the digital transformation of natural historic collections is not optional. It is important. It is essential for science. Digitalization enables cross-referencing between specimen, images, DNA sequences, ecological record, and even isotopic signature. For a mega diversity country like Indonesia, these mean three things. First, is preservations, protecting valuable information for physical loss or deteriorations. Second, accessibility, allowing scientists, educator, and policymaker to use and build upon scientific data. So we will have strong policy based on science. And lastly is integrations, connecting biodiversity knowledge across discipline and institutions. Indonesia herbarium and museum collection, many dating back more than a century, representing an irrepressible legacy. Through digital platform, we can transform them into living data, linking history to modern science. Despite these progresses, challenges remain significant. Field data collection across such a vast archipelago requires substantial resources. Many specimens are still stored within metadata, and taxonomic expertise is declining. I just discussed with our colleagues just now that taxonomy is indeed an endangered species. Many specimens are still stored without metadata, and older generation of scientists is retired. So we are in the declining process of taxonomic science. Digitalization also faces technical and instrumental barrier, from limited equipment and database to lack of standardized protocol and sustainable funding. Next, we are in BRIN, in the National Research and Innovation Agency. Next, recently launches an initiative called Terrestrial Biodiversity Expedition Platform, where big expedition composed of researchers, students, and local experts explore biodiversity rich gaps area in Kalimantan and Sumatra. So why we goes to those both area? Because we found that from the collections data that we have, that those area is still has less biodiversity informations. First batch is goes to Kalimantan and Sumatra, and second batch goes to Maluku. Next, second initiative is biological structure platform. This program integrates specimen collections, molecular analysis, and digital archiving under one coordinate system. The goal is not only to produce scientific publications, but to ensure that each specimen collected today will continue to contribute knowledge tomorrow through well-curated, accessible, and connected database. Next, research and innovation lie at the heart of these transformations. Modern biodiversity science is increasingly interdisciplinary, combine field ecology, molecular biology, informatics, and policy research. Through this effort, we aim to create a coherent national biodiversity system where collection, field observation, and digital data are integrated and standardized. digital herbarium platform, accessible database, and AI-assisted imaging recognitions are helping us accelerate species identification and improve data accuracy. This technology allows researchers in Indonesia and around the world to collaborate in real time, connecting a specimen record from different institutions into broader network. Yet, behind every innovation lies a fundamental principle, which is collaborations. We need partnership between museum, herbaria, university, governments, agency, and local community to ensure data integrity and sustainability. Next, this is the end of my presentation. So, colleagues, Indonesian biodiversity is one of the most complex and beautiful expressions of life on Earth. But its richness can only be truly understood and protected if document it, preserve it, and make it accessible. Our herbaria and museum collection are not passive archive, they are active instruments of discovery. And through transformation, we can extend their reach beyond physical wall, connecting scientists, students, and citizens across the world. So let us continue to strengthen our collections, invest in data infrastructure, and collaborate across discipline, ensuring that Indonesian biodiversity, from its forest to its microbial world, remain both visible to science and accessible to the future. Thank you very much.
Okay, thank you very much for Dr. Ruli for the presentation. So from this presentation, we heard that our beautiful biodiversity and also our rich biodiversity should be protected, and also we have to conserve, and then how to conserve based on the scientific data. We have to keep it in, at least in as a specimen in our herbarium and also in our museum actually. So after we listening the presentation that how important we record the biodiversity data as the specimen, I would like to share our experience managing the herbarium and museum in School of Life Sciences and Technology. So probably after this, I would like to present my, not my, but our Herbarium Bandungense and Zoological Museum of School of Life Sciences and Technology in Bandung Institute of Technology. So today, I would like to introduce that our herbariums, maybe next presentation. Yeah, our herbarium are established actually as the second oldest herbarium in Indonesia. And regards to those person on the slide, so our herbarium can be established, and our museum as well can establish by this collection of the specimen of the researcher from Netherlands, from America, from many countries, foreign, and also from Indonesia as well. So here our late and previous researcher, there is Pak Didi, and then Pak Undang, Pak Daru, and now Pak Ama, Pak Joko, and Pak Juandi as well. There are Indonesian researchers who contribute a lot to our herbarium and also our museum. So our herbarium and museum, as the oldest herbarium and museum for the university, we record many specimens, not only from Indonesia, but also from many foreign countries who send the specimen to us. And the establishment of our herbarium, also we will send our gratitude to Fons Tennis. There are a lot of collection from him, and also Max van Balgoy. So thank you very much for creating some system and also the specimen to us, and also Popta as the one of our founder of the herbarium. Next. Yeah, I would like to introduce our collection. So those specimens that's shown on the slides are the oldest specimen in our herbarium. So for the plants, the oldest specimen belongs to Drosera rotundifolia from 1868 from Austria. And for the Zoological Museum, we have the bird collected from 1909. So this are our oldest collection. We still keep, and only certain people can see and all can touch, and not only all the visitor can come and touch the specimen. So we have to keep our specimen safe for sure. For the collection, we have the fossil collection. So now we are trying to collaborate with our colleagues from Geological department. So they keep the specimen, the fossil specimen, some in our herbarium and also the Zoological Museum. And then we have the dry collection as well, wet collection, and then in offset, skin mount and animal skeleton for the Zoological Museum. And then we have fossil, ethnobotanical artifacts, solarium, and seed collection as well in our herbarium. The next slide is, yeah. In our herbarium here, the total specimen is around 19. So actually this herbarium just moved in 2017 from our main campus and then moved to our second campus on that time. Yeah, as Ibu Ruli said, the digitalization is important. So we lost many specimens actually on that time. So we lost the record, and then these specimens are safe until now. So we can add from 10,000 collection on that time, and then we add many collection now until to 19,000 specimen. So here from the specimen from the solarium and until the seed bank, and also some of the fossils, some of the artifacts we have in here in our herbarium. And next. Yeah, and also we are very lucky to get the type specimen. So from our collaborator in Breen and some collaborator in many countries send the type specimen to us. So now we have holotype around 21 specimen, the isotype, lectotype, and I think it is a good things for our herbarium, so we can have the type specimen in our herbarium. And then in our museum, actually, in the next slide. Okay, we have many collection about herpetofauna, and also we got the specimen from the Ministry of Forestry of Indonesia for the Panthera, and then we got the animal skeleton and also dry collection as well. The next. And here, as the university herbarium and also the museum, we have obligation for the three pillars of higher education, for academic, for research, and also for community services. So in academic, so actually our student from the main campus to the second campus to the third campus has course for biosystematics and also the taxonomy, plant taxonomy and animal taxonomy. We have for each student in each study program. So specimen from herbarium and also from the museum, every semester should be moved from herbarium to the main campus and then to the second campus. And it is quite challenging because our specimen are fragile, but we have to do that because the student have to touch, we have to measure, we have to feel about our specimen. But that is one of our challenges actually in here practical class, and also sometimes students from the summer school program want to know about the Indonesian diversity. They visit our herbarium and visit our museum, and then in our museum, the tiger is our iconic. So everyone wants to taking picture in front of the tiger. And then, yeah, our curator, this is Mr. Ganjar and also Mr. Arifin for the curator of Herbarium, working alone in each herbarium and also museum. So they have to accept student, visitor, doing research and also doing helping us in academics. So very busy actually. And the next slide, yeah, we have activity in research. Our curator are very active in writing a journal, helping researcher to write a journal and then helping them to have the field experience. So Ganja working mostly in herpetofauna and Arief in the plant. Now working started now with the recording the alien species and the new record of some species and also finding some new species with our collaborator in the other institution. And for community services. So we mostly got some visitor from kindergarten until the expert should come to our herbarium and museum. And that's also challenging because many children want to touch our specimen. And then, yeah, we have to keep them so they won't destroy our specimen. And the next slide, please. Yeah. And then we have to give some awareness to the school, the children about Indonesian diversity as well. And sometimes we are doing expedition to record many plant specimen and also the animal specimen for the community services. So this information mostly we wrote in the poster or in the flyer, or we come to the school and then we describe about the diversity. So we hope with this activity we can reach the community, we can reach the children and aware about our Indonesian diversity. So actually, managing herbarium and museum is quite challenging now because, yeah, we have, yeah, as I told before, our specimens are fragile. So we have to taking care really well to our specimen. But in the other way, our specimen are needed for many students and some researcher and also some visitor to come to our herbarium. So we have to choose which one who can touch, who can access easily. And then the other challenge are the specimen is only can be observed by certain people. And this phenomenon actually hit us during the pandemic of COVID-19. So students have to study about the biosystematics and also the plant and animal taxonomy, but they cannot come to the school. They cannot come to our university. So only we have to find how the way how the student can access our herbarium. And it is quite challenging also for long distance education. So many lecturers from the other university actually come to and then contact us. They want to see our specimen. Actually, loan is possible. Loan is possible, but they want to they want to find the catalog. They want to know our herbarium and museum, but they cannot access our database actually because that our database collection still are in our local connection. So by knowing Dr. Ruli, knowing Arief, and then knowing my colleague Fons, we are now trying to make a collaboration actually to find the opportunity how our biodiversity specimen, how our record can be broaden and then how our specimen can be used with many people. And then we are trying now to renumbering and barcoding specimen. And because it's almost 20,000 specimen now, there are specimen in our herbarium and museum. And then we are trying to digitalization because doing the digitalization, we have to we can broad our access actually, and then but back to the challenge, finding platform how to find the suitable platform means easier to manage and easier to be accessed by people. It's quite challenging for now. And then we will find how we can establish the web-based or the apps for the digitalization of the specimen. And then computerized model now, I think nowadays is quite famous, and then we want to replicate, we want to find the model so many people can reach our specimen, many people can know about our the record of our specimen. And also virtual herbarium and museum nowadays, our colleague from the design department also trying to make some virtual herbarium and museum during COVID-19 is really helpful. So they can visit our herbarium by the virtual tour, and then it's useful for our students. And when I saw in some herbarium and some museum, the hologram technology is quite interesting. So at the end of my presentation, I think I want to show the our dreams. So yeah, it's, I think, yeah, because we are not still renumbering its hologram, it's possible, but I think yeah, it's still far from our dream. But I hope with this collaboration, we can make, at least we can make the comprehensive database of the collection so many people can access our specimen. Many researchers can know what kind of the specimen that we have, and then easier to make a collaboration for research, both research and academic. And then we want to many people Indonesia, both and foreign country, also easy to access the specimen collection. And then we want the technology can improve the understanding of biodiversity from genetics to the ecosystem. We want to everyone can use this as the scientific database, and for sure at the end we want to our specimen can use not only now, but we want to we want to use this database, the specimen for education for tomorrow. So that's all presentation from me. So thank you very much for listening. And the next presenter is Arief here from the Flora Fauna Indonesia. He has many experience conducting research in the field and taking a lot of specimen from field and our endangered species probably because yeah, not so many now as the plant taxonomies. And then we will hear our presentation. Yeah, please, the stage is yours.
Okay, Dian, thank you very much for the time. Okay, I will tell the story about the from herbarium for actions here. Okay, I will try to make it in time. So, okay, my name is Arief Hamidi. I'm work in Flora International, but also here I representing the Forum of Fauna Indonesia or APLI. Okay, next. So, small introduction. Fauna Flora is an international NGO working for natural conservation and also research. In my division, I work in plant conservation and restorations to save endangered species, plant species, especially trees. Then, next. We work in 6 sites in Indonesia, covering in Sumatra, Kalimantan, Sulawesi, and also Papua. Next. Now, while the Forum Pon Langka Indonesia or FPLI is networks or also national forum that consists of various experts, practitioners, professionals, researchers from various backgrounds as well, even also from private sectors, community members, etc. And now we have around 200 people join with our initiative. We have vision to save the threatened trees in Indonesia. Next. Yeah. Then our species, 3 species in perils. How many species we have that the 3 species is especially that still exist, and how many species is threatened? Next. So previously, as Ruli mentioned, that we have Indonesia as 31. Oh, sorry, previously. So this is we are in the era of the sixth mass extinctions. That plan itself, yeah, based on IUCN, we already have the globally. There's already 133 plants has been extinct, while in this paper, 129 of plant species has been extinct. So the six mass extinction is happening. And how with Indonesia? Next, as Buruli previously mentioned, that Indonesia has 31,000 of plant species. 6,000 of them is trees, and but yeah, on 31,000, that's only 6,800 plant species that has been assessed of their threatened status. So there's a lot of species that has been assessed yet, and yeah, among this 600,000, um, almost 1,500 is threatened plants, and 1,000 and almost 1,200 is tree species. So a lot of species of tree species is threatened by extinction. Perhaps several of them may be extinct while we don't know about that, out of our unaware. One species has been extinct in Indonesia that are at Ruliyana, and we don't know how many species that actually extinct out of our aware. So yeah, next. Yeah, but most of the species has been assessed as I mentioned before. Also, most of species that perhaps undiscovered yet, as we look at here, this is in Indonesia. That's a lot of big islands like in Sumatra, Kalimantan, Sulawesi, and also Papua has quite low what is called just species richness index or density, perhaps. Like in Sumatra, there's only 1.4 species per 100 square kilometer, which is low rather than Java, which means that perhaps a lot of species doesn't discovered yet, and this potentially new species is quite high in Indonesia. But again, we found the paper that say that 3 to 4 species that undiscovered that already threatened. So again, even though this unknown yet, but they're already threatened to extinction. Perhaps there's a lot of species that already extinct without our known about this existence. Next, so all of species also, yeah, there's several species we try to conserve. FPLI and BRIN has published SRAC or Strategy and Conservation Plan Trees on Indonesia. There are 12 species priority national before we have an idea about how many trees are in perils. So next, we found that a lot of species, numerous of them are really threatened to extinctions. I'd say in Java, there are 4 species like tree of Syzygium, has category are CR, critically endangered, and PE. PE is possibly extinct. We don't know about that extinction, and we have all of species of like this. Like example, Syzygium christianum there, last seen is 1869, like several hundred years ago. So the only information of this of the species where they are is only in this herbarium. So herbarium is very important. We have no information about the species elsewhere. So yeah, we rely on this herbarium, and we have a lot of species of like this in Indonesia. Um, yeah, we have FLE and we also have released the endemic plant list of Java. There are consists of like 650 species of plant, 150 of them is trees, and most of them we are unknown on their locations. So in East Java, and then next in Sumatra, also there we have just found 2 of them, Grasnia Klabang and the Asia Tasmanii already found. The other is still unknown, still mysterious. We call it species mysterious because we don't know whether they still exist or it's already gone extinct, and we don't know how many species already gone extinct perhaps. Then in Sumatra, and then also in Kalimantan as well. Yeah, a lot of species like this. And then Kalimantan, and then in Maluku, also Sulu Island. Next, there's a lot of species. This is some of them. Most of them are unassessed yet, because even though we assess, we have no information about that. And next, also in Papua, which is now known as the richest island in the flora in the world. So as we can see here, that there are a lot of gaps there that covered for flora exploration. So there are a lot of potential for exploration. But Papua also next, Papua also has uh, so much species in Peru as well. So many species are undiscovered after its first publications. Next. Okay. Next again, this is some several species, and also this is the CR species. Next again. Now, after we know the species, then we do explorations. Next, yeah, to find out how many species still exist, and which species we have to conserve. For the flora exploration, since 2015 until now, in 6 main landscape, and we found, yeah, we record 90 species of 92 species of RTE, rare threatened and endemic species. 28 of them become our conservation program, and then, yeah, at least like 12 species in Southwest Papua is new to science, because there's no match with any herbarium for the characters, and also in description as well. So yeah, we determined that that several is just new species. Also, FPLI also do exploration to find their 12 species, and from 2020 until now, we trying to find out about the species that mysterious species, whether they still exist or they're gone extinct. Several species has been rediscovered. Most of them still mysterious. Next, then, yeah, this is where we start. As we know that information about the mysterious species is unknown. No information outside, where the only information is only in this herbarium like this. So this information about where this species found, its habitat and its character is very valuable for us. So from this information from herbarium, next to yeah, to its finding. So with long time of searching, some species finally we found them, and yeah, it's happiest moment to find it. That's at least to know that they still exist and not gone extinct. Yeah, but several of them are still missing or not rediscovered. Yeah, several in Java, like there's Media javanica or the Asia comentalis. We didn't found it yet. Several of them, like it's already discovered, like Casincordes tudorcii, Diasia pugerensis, Garcia klabang, and 4 species of Cryptocoria in Papua, like that, and yeah, like 15, maybe 15 to more or less species have been discovered out of hundreds of them. Next. So after we found the species, we try to monitor them. We make plot permanence for them. Next. Yeah, this we want to know the population, how many population, and is this still safe or not. Next, so after population study, also we reassess them, to ratify their conservation status or threatened status. And next, we also do some actions. Yeah, next, some actions like several of this. We do socialization to every so much audience. We doing roadshow to every campus. We also to stakeholders to yeah, research stakeholders, we prioritize the species to be conserved, and then we do small actions, inviting youth organization across Indonesia to do small actions. We initiate like a small grant for them, and we also invite them to collaborate for monitoring species, and we also collaborate with campus as well. 18 campus and 8 local communities or NGOs has been collaborate with us. Next, perhaps, and this is our campus network. We collaborate with campus that near to the habitat. So, well, actually, we are very open to collaborate with every campus. We are keen and very happy to collaborate with every campus, especially you have herbarium like ITB, IPB. Yeah, we are really keen to work for that to save our species in Paris and to find out how many species, and at least to answer that how many species are really extinct in Indonesia. Hopefully so little or none again to extinct, but we have to find all of them. Next, after we collaborate campus for to do some script. Also now improve research about the understudied species. So you also work with local communities to protect these species in that habitat through smart patrols and monitoring. And then next, we also trying to improve their populations. Here is an example: the Bandungensis, in the below. As like 2 or 3 years ago, we never heard about this name of species. We never know about this existence, and then we found them, and now we work collaborative with University of Jember and local community in there. We can improve its population now, and now it can become one seedling species that can be planting. Next, perhaps. Planting, and next year we have a massive movement to planting in national tree planting of the species, the first in Indonesia, and we will do it in repeating it again in next year. So from herbarium, then we can save species in place. At least we know that's how many species first, and now we can save them. Okay, perhaps the time's already up, so maybe I'm going to here. Thank you very much. I'm coming back to Dian.
Okay. Thank you very much, Arief. So very interesting when Arief can find some we called extinct, but now finding in the nature again. So that's why the importance of information from our record of the herbarium and also the museum can be used to track the availability or the existence of the species. So now we will listen from Holland. Our colleague Fons, can you try to? Hello. So from Netherlands, we have Fons and also his students, and also one of the colleagues from the naturalists will present about how the technology and what have they did with the specimen. So we will listen to the presentation from Fons, Jans, Leo, and also from Angelica. So Fons, the stage is yours. Please. Hello. Oh, Font is missing.
I believe so. He might appear in a few minutes.
It's missing actually. Font is still on the list, in the list, or no, missing?
Lou should probably also be here. They're still online. They should probably just be put on the stage.
Yeah, maybe we have to put him on the stage, perhaps.
Yeah. Or we're trying to contact, but if there is no And also Lou is missing, or still here.
I think Lou is missing also.
Yeah, Lou also is missing. The internet connection, or?
There we go.
There's Lou.
Oh,
I'm still here.
Yeah.
okay.
Fons. He
But
was
Fons.
here just a minute ago, so maybe he disconnected briefly.
Yeah.
I'm happy to start if you want.
Yeah. Can we
in
start with
period of
Lou,
time.
probably?
Sure.
Just to keep our time. Okay, thank you, Lou. The stage is yours, please.
Yes.
So Lou or me? Lou, you will start.
Me.
Lou first. Yeah.
Go ahead.
Okay.
Yeah.
Could you firstly find my slides?
Yang presentasinya Lou.
Fons is asking me to maybe let Angelica start, because I believe she has a lecture to teach at 11.
Oh, okay. Okay, then we change the situation. Okay, then we change the order, and then please, Angelica, the stage is yours.
Perfect, thank you.
Oh, thank you so much. Yeah, can you please put the slides up? Do you want me to share screen, or I thought my slides would be up?
From the organizer, probably.
Yeah.
Okay. We are trying to share the slides. Okay. Okay, this is your presentation, Angelica, please.
Thank you so much. Well, thank you so much for the invitation. It was a pleasure to hear the previous speakers talk about collections and the importance of herbaria and specimens, especially now that indeed we have taxonomists at the brink of extinction as well. And we've been thinking a lot about this and what can we do to preserve this expertise and this knowledge. And we've come up with a few things that I will mention throughout this talk. I will give you an overview of what we are, who we are, and the kind of projects that we're undertaking that are large-scale projects that perhaps will also be of interest to you. So next slide, please. So Naturalis has about 200 years of history. We are the biodiversity center of the Netherlands, and we have approximately 42 million objects in our collection, more or less, with approximately 6 million plants in our herbarium. We are a merger of previous herbaria from Wageningen, from the University of Leiden, and from the Zoological Museum of Amsterdam and the Natural History Museum in Leiden as well. And the mission of Naturalis is to understand and characterize biodiversity, not just in the Netherlands, but also abroad, especially in tropical regions, including Southeast Asia and the Amazon. And I will go into this in a little bit. And we have an area of research. We're approximately 200 researchers with a lot of guest researchers on board as well, and 8 research groups focused on various topics. I am the head of the tropical botany department, in which we're focused on tropical plants in particular, but also a little bit of the Netherlands plants. And recently, Naturalis has undertaken large projects, which I'll mention in a minute, regarding not just our collections and how they are already, but also how can we collect differently and how can we plan ahead in a standardized way for the future. And I will go into more detail in a minute. And as I mentioned, we've been in various areas of the world collecting and trying to catalog biodiversity. We also have established collaborations with international organizations and international databases so that anything and everything that is done in Naturalis can be digitalized and be accessible throughout the world. I will mention that in a minute as well. Next slide, please. Yeah, so I mentioned before, approximately 150 to 200 researchers, 8 research groups. We have affiliations with Dutch universities and also universities abroad, including Indonesia. to host students. We don't have a graduate program, but we're actually affiliated to all these other universities to give the degrees in graduate education. And we have collaborations not only with academic institutions, but also companies and also governments and NGOs and other types of stakeholders. We, of course, have massive outreach with millions of visitors per year. But most importantly to us today are the initiatives that have to do with collections and the way that we sample biodiversity. I invite you to go to the links that I have in these slides to get further information, as we don't have that much time today. But I will try to give you a brief overview. Next slide, please. So there's something that I did want to highlight is the laboratories that we have, and this is because we are open to collaborations and training, and there are several facilities that may be of your interest. We have approximately 20 different laboratories across morphological tools, geology, microscopy, next generation sequencing, including an ancient DNA lab in which we're able to process, for example, ancient soil samples or paleobotanical samples. We have the facilities to do that. And we welcome students and collaborators throughout the year to work with us and to come and use our facilities and also learn techniques within these laboratories. We also have an informatics team. Recently, we opened a new department where we're trying to optimize anything that has to do with bioinformatics, including all the AI algorithms that are linked to computer-related processes. And I invite you to look at the website to see if there's something of interest to you, and you can get in touch with me to talk about how and when and under what agreements we could have you visit us and maybe collaborate on a project. Next slide, please. But the most interesting that I wanted to highlight today to you is, in addition to doing collections in a traditional way and going out to the field, sampling something, pressing it, and including it in a herbarium, of course, digitalizing. Recently, we've been wondering, can you also listen and record nature in different ways, such that a single specimen has multiple layers of information, and not only the physical voucher or the physical specimen, but also how it sounded in nature, how the context looked like, and more deeply, its DNA, both at the individual level and also in communities. So we've set out two large projects. Next slide, please. In which we have built infrastructure specifically to address these two projects. And one of them is Arise, and the other is Identity. And Arise, the main goal is to sample all multicellular organisms of the Netherlands with a single genetic barcode. This is very specific to certain barcodes for plants and certain barcodes for animals, and to have that information. On top of that information, also have, like I mentioned before, sound, anatomy in more detail, morphological detail of some of these specimens, videos if it's possible, and use AI to integrate all this information. And Identity focuses mostly on the smaller microorganisms, both bacteria and fungi. And Identity, unlike Arise, is not a single barcode, so a single DNA signature, but in this case, we have environmental samples, and we do metabarcoding with many genomic barcodes or codes to be able to characterize everything that is in a sample in its complexity, for like I said before, like a soil sample. Next slide, please. Next slide. There we go. And so I just wanted to mention Arise in a bit more detail. This is the beauty and the importance of these projects is not only the scale of how many samples we want to be able to include, but also to have very clear protocols that can be replicated over and over and that can give similar metadata. And this is very important because then you can do comparative studies. And for now, we're focused mostly in the Netherlands. And the protocols that we have developed from the initial sampling to the AI processing, we're still developing them and we're still building on them because it's not easy to standardize across all biodiversity. But the idea is that it's not applicable just for a place like the Netherlands, which has less biodiversity in general than biodiversity hotspots, including Indonesia. But the idea and the goal is to also be able to transfer some of these techniques and some of these protocols to very complex and biodiverse places like Indonesia. So this is something that we can talk about, and it's something that I am specifically exploring for tropical plants and tropical regions. And so surrounding a species in the context of Arise, we have traditional taxonomy based on morphology, based on photographs and a digitized version of the specimen. We have observations in the field. We have descriptions of the surroundings or metadata associated to the species. We also include traits that have to do with behavior or any additional traits that we might want to add about the species. We have, as I mentioned, a DNA barcode. Sometimes we include a location via a radar. We have sounds of the species and the images. And this all goes into one specific archive for that particular species. Next slide, please. And in order to do that, we have 5 different teams of people, and you are more than welcome to explore the website of Arise, where there are some videos, some additional materials. People can volunteer through an app on their phone to add some of the data for these specimens. And these 5 teams are divided into very specialized sections of this pipeline. And the first one is how to collect and how to manage the samples from the beginning, so that, again, as I mentioned before, we can actually compare what comes out of that sample. So this includes the way you actually collect sample, the way you store the sample, how it gets shipped to our facilities, how it gets stored within the facilities, for example, if it needs to be frozen immediately, or if it's going to be conserved in ethanol, or if it's just simply going to be photographed. All of this is part of the team that does the collection in the first place. And we have, we adhere to international protocols like MDS, which is a museum-based standardized protocol. And in every step, we have our own guidelines and protocols, but also we try to pair up with international and recognized guidelines, so we can compare not only within the Netherlands and within the various samples that we're processing, but also with other large initiatives that are similar to this one. The second step is, like I mentioned, in part storage of the sample itself, but also storage of the data. And for example, we follow IRODS, which is the Integrated Rule for Oriented Data System. And we have a partnership with Biobank and a biobank in itself. We have also, of course, the bioportal, which you are free to consult at the moment. The bioportal is the main hub of Naturalis, where we have most of our, or a good portion of our specimens already digitalized. And recently, we consolidated a collaboration with GBIF, in which all of our collections are also in GBIF. So this is a platform, a global biodiversity information facility, where you can also find these specimens. And the idea is that everything that gets collected immediately is linked to databases that are accessible not just internally, but also worldwide. Then the third part of the team, which is important, is the sequencing. And in that also there's a digital sequence information system that we follow, so that the sequence, again, can be used throughout the world using very clear information. And we store this in a BioCloud. This is the fourth team. And finally, we make it accessible, like I mentioned before, not just the biobanks, but also GBIF and other platforms in which the user interface is friendly, so people can actually search things and look up their favorite species or simply discover new species. Can you, the next slide, please. And so this was just a brief overview of one of the several programs that we have ongoing, but we are also thinking on how can we build on this, because these are large infrastructure projects, but we also want to make things a bit more agile in the field, for example. And so we're currently working on bringing the guidelines and the protocols that we've developed for Arise and Identity, which are linked to a physical infrastructure within Naturalis, but bring it out into the field using observation towers. So these are standalone towers that we can use to sample biodiversity in the field. And we're also thinking about how to integrate knowledge that is already there in local communities and co-create biological new indicators and new ways of collection. This is through Wisdom. This is a tool that we're currently developing. And finally, we're adding other sources of information to the specimens we already have in the collection by exploring sound and infrared light spectroscopy to have additional taxonomic traits. And I'm happy to talk about this with anyone that's interested later. Next slide, please. And this is the last one. Yeah, thank you very much.
Thank you very much. We have to learn a lot from naturalists probably to improve our herbarium. Thank you, Angela. Yeah. For the next presenter is Lu. Yeah, okay, the stage is yours.
Thank you. So it was nice to hear all these people working in the museum and herbarium collecting a lot of samples. And now I would like to introduce how we analyze these samples and how we make use of them to do some classification. So today's topic from me is the pollen classification. And by the way, I will briefly introduce myself. Now I'm an assistant professor in the Imaging and Bioinformatics lab led by Professor Fons Van Verbeek. So I'm now a colleague of him, and in Leiden Institute of Advanced Computer Science in Leiden University. Okay, next slides. So in this study, we especially focus on 3 species and also from the 2 genera. So I will introduce them. These are from Urticaceae family, and one of the genus is called Paritalia, and the another genus is called Urtica genus. And inside of this Urtica genus, we also include another species called Urtica membrianaceae. So the reason why we try to look into these 3 species from 2 genera is because the pollen morphology is rather close, similar. However, the allergic level is very different. For example, Paritaria is very allergic. That means it can cause a lot of hay fever symptoms. However, Urtica is not allergic at all. And the reason why we incorporate this Urtica membrianaceae is because it's an exotic Mediterranean species in the Netherlands now and caused by the climate change. Next slides. Next, please. So when we're looking into the pollen species, we see so what we are seeing is a slide scanner, a 2D slide scanner in the sub graph of A. They are 2D. And the first step we need to do is to crop them. So we try to find individual pollens from the slide scanner. And I also want to mention that these species is being collected freshly from the nature and also from the herbarium in the Naturalis Museum. And when we see the subfigure B, we see they are not just the 2D images, they are actually 3D images. We collect 20 slides in the Z direction. However, when we try to use machine learning or deep learning models to analyze or to do the classification, it might be too much or computationally expensive to use the whole 3D data set for the processing. Therefore, we try to reduce the space, but still preserve important information from this 3D space. And what we do is to do projection. So in the subgraph C, you see different projection result in a 2D space. The first one is the standard deviation, and the middle one is the minimum projection, and the last one is extended focus. So we try to do the projection from the 2D space into from the 3D space into the 2D space, but try to reserve as much information as possible. And the last step in D is trying to make sure all these pollen image is in the same space. So because when we throw these image into the machine learning or deep learning models, we need to make sure they are in the same size. In order not to destroy the morphology a lot, we do the padding. So we don't touch the pollen, we just pad the rest of the area. So that is the last step for the pre-processing. Next slides. So when we look into these three projections of these three species, we can see the Urtica membranaceae, the C, is the most distinct one. So we can easily distinguish this one with regard to Paritaria and Urtica. The other two is more difficult to identify, and it's even very challenging for a expertise. So what we do, we collect about 6,000 pollen images, and we have an even distribution for 3 classes. So for each class, we have about 2,000 samples for training the classifier. Next slide. So we try to use all kinds of available strategies to do the classification. So we can use machine learning strategy and also the deep learning strategy. For machine learning method, we need to extract the features from the image first. So these are handcrafted features. These are different names. Just let you know, these are the features. We try to extract the texture features from the image. And the dimension of the feature is also varied by different filters. But at the end, we will have about 4,000 features multiply 3 because these are 3 channels. We have 3 projections. And with these features, we will make use of classifiers, including the support vector machine, random forest, a multi-layer perceptron, and also the ADA-post classifier to do the comparison. Next slide. So in the machine learning strategy, we also could incorporate some hierarchical structures we have in our 3 classes. What we observed that the Urtica membrana is morphologically more distinct comparing to the Paritaria and Urtica. Therefore, we try to bring them into a hierarchical tree. And in the first layer, we have label 3 and a grouped label of Paritaria and Urtica. We try to firstly train a classifier to distinguish these 2 labels, and then we will train another classifier in level 2 to especially focus on label 1 and label 2, the Paritaria and Urtica. Next slide. Of course, we not only have machine learning method, we also have deep learning method. So here we try to bring in some representative deep learning models. These are all convolutional neural networks from AlexNet, BGG, and ResNet. So the AlexNet is proposed in 2012. It is a layer with 5 convolutional neural networks, and the BGG is a bit deeper. They are about 16 to 19 layers of convolutional neural network, which has been proposed in 2014. And ResNet is proposed in 2016. They are much deeper. They are specialized by its residue blocks, which can construct the deep learning model much deeper. So they can reach to about 100 layers. But for now, in this paper, we especially focus on the ResNet 50, which is 50 residue blocks we included in the structure. So next layer, next slide. Then we will look into some performance comparison. I try to highlight them to make it clear. So when we go through all these machine learning methods, looking for different classifiers, we see the support vector machine give us the best performance with the F1 score with about 0.913 for the tenfold cross-validation. Next slide. When we also incorporated the hierarchical structure into the machine learning method, so we try to train two classifiers in different levels, we see the support vector machine also perform the best with a F1 score about 0.941. Next slide. And for the deep learning method, before we checking the numbers, we firstly need to check if the training goes well. So we see the performance of these. So the yellow lines and orange lines is the accuracy, and the blue line, gray line is the loss. So we see whether the training and the validation give a similar accuracy and a loss. So otherwise, they would be overfitting or underfitting. So here we basically check the performance with different methods and with the increasing of the epochs and the batch size. Next slide. So after that, after we are sure that the training step goes well, we compare the result, and we see is the ResNet 50 give us the best F1 score about 0.994 for the tenfold validation. Next slide. So in the end, after we compared machine learning and deep learning, the ResNet 50 give us the highest accuracy or F1 score. Therefore, we want to look into what kind of data they do well and what kind of data they fail the prediction. So on the first line, we see all these successfully classified pollen samples, and the second layer are wrongly classified ones. But we can also see on the second layer, these pollen is already, the shape of the pollen is not very healthy. They have some concave shapes on the edge. Okay, next slide. So this is give you an impression on how we analyze them and how we make use of the pollen image to do the classification. But this project is continuing. Now we have stained pollen still being imaged in the slide scanner, and we use a similar strategies using the projection from the 3D space, the standard deviation minimum and max projection. And using these pollens that we can get, then we can use these knowledge and transfer these knowledge from herbarium and to actually identify pollens in the air. So that is the whole point of this project. Next slide. And I would like to thank our collaborators. Barbara Gravendeel is a professor in Naturalis. Now she's the scientific director in Hortus. It's another museum, especially focused on plant species. And the following is two of her PhD students, Marcel Polling and Nemi Dost. We also have Professor Fons Van Verbeek, who is the head of our lab, and with me and our PhD student, Chen Li, who is involved in this pollen classification project. Thank you. Next slide.
Thank you, Lou. It's very interesting. So from the fresh specimen, and also from the probably from herbarium, we can also use. We can try to learn how to the deep learning working with our specimen. And also the interesting things is you work with introduction species. So related to our now concern about the new record of the invasive and also introduction species, probably because some of the pollens can cause allergy to some people. Thank you very much. Very interesting information. So, Jens, are you ready now? Okay. Are you ready? And then the next presenter is Jens, and the stage is yours, please.
Thank you. There should also be some slides ready, if that's okay. If not, I'll keep it really brief, I guess.
Yeah. Heavy rain here, so probably the signal is not go with the rain. Since we are waiting to preparing the presentation.
Perfect. Thank you.
Yeah.
There we
Okay.
go.
Yeah.
Oh, this is Fons' slides, but we can let Fons go first. It's fine.
Wait.
Can I be heard?
Okay.
Yes.
Yeah.
I
Your voice.
was somehow kicked out, but I'm back. So basically, I'm happy that Angelica was able to start the presentation of Leiden. And Naturalis is a Leiden institute, but it stands alone, as she explained. Lou did some very nice about herbarium species and the collection of herbarium species next to fresh species. Jens will now talk about some work that he did in Indonesia on whole genome sequencing of an endemic Indonesian fish that might be a very good model system to us, but it's also interesting to see how he used that to use phylogeny of in relation to other species. So he'll probably make it very brief, but I would like to give him the opportunity to explain what he did very recently in Indonesia in the Yogyakarta region with UGM University.
Thank you. I don't know if there's... Yes, there we go. So yeah, this is just a very brief presentation. The idea was to make it a little longer, but we're running out of time, so I'll be very brief. I don't know if I can skip through the presentation myself. I can. Nice. So in this context, there's a very short personal introduction. Then I'll introduce two products that I'll quickly be discussing. The first one is, like Fons just said, about an Indonesian fish that we did some genome sequencing with. And the second one is the future work in the field of biodiversity conservation. So first of all, my name is Jens, as just mentioned. I did both the bachelor's and the master's in bioinformatics in Leiden, where I learned from, among others, Fons and Lou. And now I'm actually allowed to be a PhD student in their research group, which is an honor, meaning that we can further our research into the bioinformatics field. And in this case, that's a very large field where we apply computer science to biological problems and data sets with, among others, thing in genetics that we'll be discussing first, which is the Rasbora project, and then research in biodiversity. So first of all, the Rasbora project. At Leiden University, we used the zebrafish, the Danio as model organism for many different biological problems, such as research into cardiovascular disease or the immune system. But for research into human tumor cells, it's unfortunately not really well suited because its temperature conditions under which it thrives are just too low, because human tumor cells grow under 37 degrees Celsius, and zebrafish usually die under those temperatures. And the solution for this, the possible solution for this problem, actually came from Indonesia, where a Dutch scientist called Peter Bleeker went in the 1860s to do research into the different types of fish that are available there. And he found the Rasbora latrostriata fish, which should be a family member of the zebrafish, only it is able to live under higher temperature conditions. And as such, just this year, we set out to Indonesia, to our partners at the UGM Faculty of Biology, in order to do research into this fish and try and develop it into a model organism, where the first step would be to create the draft reference genome. So we're basically mapping its genetic information. And the goals of this project were twofold. First of all, we wanted to create an annotated reference genome for the Rasbora latrostriata fish. And second of all, we wanted to use the created reference genome in a synteny and a phylogenetic analysis in order to ensure that it is indeed family of the zebrafish like we expected as a hypothesis. And the collaboration was really fruitful because it allowed us access to certain resources. The team at UGM currently has set up the Integrative Genome Factory, which has its own sequencing devices, such as, for example, an Oxford Nanopore Technologies Promethean 24, which we used to great success during the project. And Leiden University provided access to the national supercomputer in the Netherlands called Snellius, which is managed by Conserve, which was used for all the computational analyses. And both of these were used a lot, as you can see in the following flowchart of the methods for this project, which I will no longer dive into in great detail, but we'll just skip to some of the results, where we can see that we successfully generated a genomic data set for this until now unmapped Indonesian species, and successfully used it to, for example, make an estimation about the genome size of the Rasbora latrostriata. We then use this data set to do some assembly, which is where we take the different types of genomic data we have and paste it together. And we improve that through scaffolding based on the zebrafish because of the estimated lineage. And then after some screening, we were able to do annotation and arrived at the eventual reference genome, which is in itself a great success and a great conclusion to the story. But like we said, the second goal was to do some phylogenetic and synthetic screening. So we did that next by comparing the created genomic regions for the Rasbora, which we can see on the top left, to, among others, zebrafish and carp. And as we can see from these two graphs, the amount of crossovers between the Rasbora and carp is greater than the amount of crossovers between the Rasbora and the zebrafish, which confirms our hypothesis that the zebrafish is genomically probably closer to the Rasbora. However, in order to further confirm this, we also did a phylogenetic analysis where we can luckily see that, that indeed the Rasbora maps most closely to zebrafish, which is a very nice result. And it shows that a collaboration between a research institute in the Netherlands and in Indonesia actually can lead to some great results. And we're still hoping to develop the Rasbora further into a model organism. But for now, this is the conclusion of the story. This is how far we got, which only keeps us to talk about the future in this case. And this is a new project that we are currently working on. That is called pattern formation through bio-inspired computer vision, or tattoos for short. And it's a collaboration between us at Leiden University and the Naturalis Biodiversity Center that we've heard much about before, where we are going to be trying to use computer science in order to do some biodiversity conservation. And I really quickly like to mention this just because I thought it was funny, that there's, of course, a very nice link between Naturalis and Indonesia through, for example, the Dubois collection that will be returned somewhere in the coming time. But biodiversity conservation is very important right now because, as Arief just mentioned, we are in an era of extinction, and predictions model that we should really step up our conservation efforts if we want to keep the biodiversity at least a livable level. And as such, a lot of data has been collected. As we've just discussed, there are a lot of herbariums that have a lot of multimodal information about, for example, tiger species, such as image data, video data, sound data, but also GPS data, data from different sensors, like, for example, temperature, and some semantic data, for example, observations done by biologists. And all this data can be nicely used by computer scientists, such as, for example, myself, to do like they did for the Flukebook project. First of all, detection, and second of all, identification of individual species, which could lead to a better conservation effort. And as such, for the Tattoos project, we are trying to do a similar thing as the Flukebook project did for whale species, but this time for a lot of different species by basing the identification on the different patterns on animal skin. So in this case, we're of course using zebrafish once again as a model organism, where we're trying to create a network that will be able to simulate the natural variation of this structural pattern on zebrafish, and then use it for individual identification of the zebrafish, and then furthermore to do population monitoring using this individual identification. And once we've furthered this project far enough, we're hoping to do generalization with it, meaning that we can eventually use it for every animal with a distinguishable pattern, such as, for example, giraffes or Sumatran tigers, for example, which nicely links back to Indonesia once again. But for now, like I said, this is a project that we're currently working on. So we're currently still teaching the architecture of this computational model, how to create such a pattern, in this case, how to fit it in. We're currently looking into an architecture called Llama, which is one of the newer AI models that uses information from an image in order to create a pattern on a blocked out area of it, like we see at the top here. And as promised, this was a very, very quick overview, a very quick walk through the two projects that I wanted to discuss with you on this conference. Thank you very much.
Thank you very much. Very interesting. So we learned from the field, from Arief, and then we learned how the Indonesian diversity current condition, and then also we heard from my side with how to manage the herbarium. And we can see that many things we can do, a lot of things, the opportunity to work with the specimen and then work with the fresh specimen, and actually how incredible diversity is. So incredible the nature form is. So we can learn a lot of things from our nature. So open collaboration with all of us. So let's work with the biodiversity. At the end, we have to conserve diversity, because the diversity can support our human well-being, actually. So we can learn many things from today's presenter. So I hope it's can deliver well to you all also. Thank you very much from our session. So I be return the stage to the organizer. Thank you very much for all the presenter and the listener, and also from Netherlands. So here is a heavy rain in Bogor. So I hope the signal is still okay here. So thank you very much and see you. And I know we are waiting for the discussion session. So we'll.
Thank you.
Thank you. Bye. Thank you very much.